Hi, everyone. Today we're going to be doing a quick overview of cellular metabolism, specifically the steps of cellular respiration, which is the coupling of the oxidation or breakdown of a fuel. We're talking about usually glucose or a fatty acid. So we're going to couple the oxidation / breakdown of that fuel to the synthesis of ATP. That's the big picture. And before we get too in depth there, we have to know the anatomy of sort of the structures of the cell that are doing this. So we're going to start on the whiteboard here, and I'm going to draw our key organelle for today, which is a mitochondrion. Mitochondria is plural. And we're going to have multiple steps going on here. The first thing to realize is that the inner membrane, which is the original bacterial membrane, if we think about our endosymbiosis events, this is highly folded. And you obviously don't need to know every single fold by heart because these are varied. These aren't going to be the same. And so they are going to be highly, highly folded like so. And on these inner membrane surface, on this membrane, this is where you're going to have the key enzymes for ATP synthesis. So let me label a few things here just so we have them for later. This area here, which you could imagine being the cytosol of a bacterium. All right, evolutionarily, it is similar to that. This is known as the matrix. Obviously, we should have a better name for that. It should be the mitosol or something like that, but we're going to call it the matrix. That is the ancient term for us. And then these folds of the inner membrane, the folds of the inner membrane are known as cristae. So these are just our basic vocab terms for today. And then out here is the outer membrane, and another term that is worth knowing is the space between the two membranes is the intermembrane space. So there is just our vocab. And just remember that if we think of the inner portion of the mitochondrion as being a bacterium, you can just think of this as being the outside world. This is a luminal space, basically, outside world space, and this over here is cytosol. I'll also note that a lot of the things we're going to talk about here are very similar, extremely similar to things that happen in chloroplasts as well, but we'll get to that later. So we're going to be talking about three different steps, sort of. The first step happening in the cytosol primarily, as well as in the matrix, is going to be the breakdown of a fuel. All right, and we're going to take a fuel like glucose or a fatty acid or an amino acid, starting out here in the cytosol, break it down to the point where there's nothing left and you're going to eventually exhale the gaseous products. That's step one. Step two is going to be a process on this membrane called electron transport chain. And the third process is actually where we're going to begin. The third process is the ATP synthesis event itself, which happens thanks to an ATP synthase enzyme, and there are thousands of those per mitochondrion. Again, this is found on the inner mitochondrial membrane, typically paired actually. But I'm going to show it in a particular arrangement. You'll see it's often described as a lollipop shape, and the lollipop end is found in the matrix. And that's going to be the location that does the conversion of adenosine diphosphate, ADP, and a free phosphate, usually abbreviated as P sub I for inorganic phosphate, so that it doesn't get mixed up with a phosphorus atom. Conversion of that to ATP. Now, earlier we've talked about that, and you've seen videos hopefully at this point about that process, but this ATP synthase is driven by a proton gradient. And if I haven't talked about this before, or you've missed a video, that proton gradient is leading from this intermembrane space into the matrix. That is the direction of the concentration gradient. And so the protons, again, are found here in the intermembrane space. That is where there is the highest concentration. And we'll talk about how we generate that in a moment. So just remember that the intermembrane space has a high concentration of protons, which you could also describe as being acidic, because it will be a relatively high proton concentration. So this region is relatively acidic compared to where the protons originated, which was in the matrix. So since the protons originate in the matrix, this is relatively basic. If we wanted to put some pHs to it, we could estimate it. This would be a very ballpark estimate. This would be a pH 8 region, perhaps, again, mildly alkaline. And then this intermembrane space might be pH 6. Again, this is a very rough estimate. Keep in mind that this is a log scale of pH, therefore this is the way I've estimated it, this would be 100-fold difference in proton concentration. If this really were pH 6 and pH 8, this would be 100-fold higher concentration of protons in the intermembrane space as opposed to the matrix. Okay, why do we care? Well, the reason we care is that this proton flow from high to low concentration, that would be an exergonic reaction, diffusing this manner, diffusing this direction. That would be an exergonic process that is going to be coupled to the movement of a motor within this synthase, which leads to the production of ATP, which we're not going to go into the details of today. What I want you to see are the steps that lead to this proton gradient. How do we get that proton gradient which drives the ATP synthase? Okay. The actual process that's going to lead to the proton gradient is a process called electron transport. And so that is done by a set of proteins found in this membrane as well. So also found on the cristae are a series of proteins. I'm going to draw it as 3. That isn't necessarily accurate, depending on your point of view in terms of membrane versus non-membrane proteins. I'm going to draw it as a set of 3 proteins here. And again, this is the electron transport chain. Now, what this is a series of proteins that, when it goes through a set of shape changes, pumps protons into the intermembrane space. We're going to have to feed that some high energy electrons. So I fully understand that this is horrifically confusing at this point. Just remember, this is going to be something that's going to drive protons into this space, and the proton gradient drives the ATP synthase. So let's take a quick step back. Okay. We need what are called high energy electrons to fuel this process. We'll get to that later. Where are those electrons coming from? Well, they're coming from fuels. So the fuels we're going to be talking about typically are the things that we eat. And so the fuels that we typically think about are things like glucose or similar sugars. Also possible are things like fatty acids, if you're running on fats, or the cell perhaps is running on fats. And the other possibility are amino acids. If you're running on proteins, which typically is not true for most cells unless they're actually starving. Now, regardless of whatever fuel you're using in this case, all 3 of these are going to lead to typically one breakdown product that is used inside the mitochondrion. And inside a mitochondrion, the breakdown product of these is known as Acetyl Coa or Acetyl Coa. This is just an acetate group attached to a vitamin. Okay, so this is Acetyl Coa, and we're going to break these things into this, and then this is going to get broken down. Bigger picture, this is basically just a 2-carbon group, a 2-carbon sugar-ish thing. So I'm writing it as 2 C's. This is a 6-carbon thing, carbohydrate, 6-carbon saccharide. This over here is a lot larger, a lot of carbons per fatty acid, and this will be a small number. What we end up doing is we end up breaking down each of these into smaller units and basically harvesting electrons. When a molecule loses electrons, that's known as oxidation. So this is going to be an oxidative process. We're losing electrons. If you don't remember that term, I'll draw it on here. Loss of electrons equals oxidation. Gain of electrons equals reduction. What we're talking about is oxidation of these fuels. They're going to lose electrons, and we're going to transfer them to the next step over there. The way to remember this, Leo, L-E-O, Ger, G-E-R. Leo the tiger goes Ger. Another way of doing this is oil rig. So oxidation is loss, reduction is gain. Okay, I'm going to erase that so it doesn't take up too much room on this board. At least I'll get rid of the reduction portion of this. And say that we're going to do an oxidation of glucose into a molecule called pyruvate. That's just half of a glucose. Don't overthink this. All the videos are going to show you multiple like intermediaries and lots of complexity. You don't need to know any of that stuff. We want to focus on the big picture. Fuel broken down into some intermediary form, okay? And as we break it down, we're harvesting electrons. So as we're breaking this down, we're harvesting electrons. This process, where we go from 6 carbons to 3 carbons to 2 carbon molecules, and eventually to our 1 carbon molecule, which hopefully you know about in our final end result here, is going to be a 1 carbon molecule known as CO2. So literally, when you break down sugars, there's nothing left in your body. Like literally, when you break down a sugar as fuel source in your body, you end up exhaling it. It's gone. It is physically removed from your body by exhalation. So that's what we're doing here, but we are going to hang on to the electrons. We're stealing the electrons. Okay, so glucose is broken down to pyruvate. That's a 3 carbon molecule, so it's 2 of these, breaking it in half. So we break a glucose in half, then we break it. We break CO2s off of it to make a 2 carbon molecule. And then each of those 2 carbon molecules is broken all the way into single carbon molecules, and that's CO2. So in the end, we start with 6 carbon glucose. We end up with a bunch of CO2s. Don't get too confused by that if you can avoid it. These 2 processes where we break down the glucose have names. The first process is known as glycolysis. And the second process is known, at least according to the College Board, as the Krebs Cycle. There are multiple names for this. You'd call it the citric acid cycle or the tricarboxylic acid cycle. Krebs cycle is what the College Board typically uses. So these are just 2 breakdown processes, glucose to pyruvate, and then acetyl Coa. Acetyl Coa broken down into CO2. These processes harvest electrons, which will be the next step. At this point, I would typically pause for questions, but you're not here, so I can't. I will quickly note that amino acids can also be shunted into this process, a different process that ends up creating generally acetyl Coa. Fatty acids can also go down to a different breakdown process called beta oxidation that also typically leads to acetyl Coa. There are plenty of exceptions. We're not going into those for today. But understand that there are multiple fuel sources that can be used to fuel mitochondrial ATP synthesis. Okay, so what did we get from this? Big picture. We do this breakdown of all these fuels. We exhale the results, so there's nothing left. Why did we bother doing that? Well, we did steal something. We did steal something from these molecules. As we were breaking them apart, we're going to steal some electrons. Okay, one more time. This is an oxidation process. Oxidation is a loss of electrons. So we're literally ripping away the electrons from these fuel sources as we're breaking them down. And there is a molecule that's going to basically take those electrons and transfer them over to here. This molecule that's an electron carrier is known as NAD. So I'll state that again. It's known as NAD, nicotinamide adenine dinucleotide. No, you do not have to memorize that. So what is it? Nicotinamide adenine dinucleotide, also again known as NAD, which I'll show with a positive sign, is basically a vitamin. This is a variant of niacin, if you've heard of that B vitamin before. This is a variant of niacin. And so what's going to end up happening is NAD is going to accept electrons from this breakdown process from this oxidation process and become negatively charged, pick up a proton. Please accept that. Don't ask too many questions or else you'll get a headache. We end up with NADH. We started with a positive charge. We're picking up 2 electrons and a proton. Just think of it as a pair of protons. Sorry, a pair of electrons. And so we end up with NADH. This is the process. Every time we do a breakdown step, electrons are harvested from the fuel source, transmitted to NAD+. This is our electron carrier. Same thing happens down here. We have electron carriers that take electrons from acetyl Coa, and these NAD+ become NADH. And there's even another electron carrier known as FAD, and we're not going to go into the details of that either. Just understand that there are multiple electron sort of carriers here. So once again, we're breaking down all of these fuels. As we do that oxidation, we steal the electrons, give them to a carrier. This is just our transfer device. These transmitters, these electron carriers, the NADs, and the FADH2s, are in the matrix, and they will diffuse to one of these locations over here, known as an electron transport chain. And they will drop off their electrons. The NADH drops off its electrons and is now NAD+ again. Okay, why do we care? Why do we care? Let me take a step back. Why do the electrons matter here? Big picture, those proteins of the electron transport chain, when I can do this simply using a half-assed diagram here, or model here, when an electron binds to this thing, it changes shape. And as it's changed shape, it's going to fling a proton to the other side of the membrane. I'll state that one more time. When an electron is passed to this thing, it goes through a shape change. So it may, if I was going to grab my props as usual, in one particular orientation, one particular shape might have a proton on one side of it. When the electron binds to this protein, some portion of it is undergoing a charge change, which is going to lead to a change in bonds. And as this goes through a bonding shift, it's going to change shape. Electron binds, protein changes shape, flings a proton across the membrane. Okay? When the electron leaves, it goes back to its first shape. One more time. Electron arrives, changing this protein's shape, which flings an H+, flings a proton. When the electron leaves, it goes back to its original shape and can pick up another proton. Electron arrives, changes this protein's shape, causing it to fling a proton. Electron leaves, returns to its first shape. Okay, back to the diagram, and we'll finish up here. So that's going to happen at these things that are marked in green. And yes, this is a very, very full diagram. The proteins shown in green are membrane proteins on the cristae, and these are known as the electron transport chain. Their importance is as they go through the shape change process, as the electron arrives at the first and then leaves to go to the second, arrives at the second, leaves, goes to the third, leaves the third, we'll talk about that next. As the electrons are passed from NADH to electron transport chain component 1 to component 2 to component 3, each of these proteins undergoes a shape change, causing protons to be flung into the intermembrane space. That's the way the protons go. And now we have the proton gradient that's going to drive the ATP synthase. Last part. The electrons go from fuel to carrier to electron transport chain, but then where? I'll start that again. The electrons in this whole process of cellular respiration start at the fuel, the glucose for example, are harvested from these fuel sources handed over to our electron carrier, which becomes NADH. NADH drops the electrons at the electron transport chain. And they go from component 1 to component 2 to component 3. But now what? This is a problem. And I'm going to highlight this area right here. I'll draw it right here. So let's zoom in on this membrane. Zoom in on this membrane. This membrane has, let's just say, 3 electron transport chain components. 1, 2, 3. Third one being the most important, or the key for this one. And we just talked about the fact that NADH is going to drop off its electrons. That's going to happen here. And the electrons are going to pass from component 1 to component 2 to component 3. And as these components get the electron and then pass it along, they undergo a shape change, which causes, very specifically, proton to get pumped across. Boom, boom, boom. Protons get flung into the intermembrane space. And if I was going to draw the location of the electrons, I'm going to use a terrible yellow marker here just to show you where they're going. Going from NADH to component 1, then they are passed to component 2, and then they are passed to component 3. Here's the last step, though. This has to get rid of that electron somehow, or else it won't be able to go through its shape change again. Remember, these proteins have to go through with the electron, without the electron, with the electron, without the electron, with the electron, without the electron, with the electron, in order to effectively pump the protons. Once it has that electron, it has to be able to give it to someone else. And there's not much else for it to give it to. There's no other electron transport chain component that is electronegative enough to actually pick up the electron at this point. There's nothing that can take it. So the only thing we're left with is the most electronegative thing in all of life. So I'll state that again. The only thing that's electronegative enough to take the electrons from this component at this point is the most electronegative thing in life. And that last thing is something that you all know about, O2. Let's not talk about fluorine. Not enough fluorine to be important, all right? Not for this class. O2 can accept these electrons, becoming effectively O- radicals. So the electrons are going to be passed off to to oxygen. And as they go to the oxygen, it becomes O- radicals, picks up protons. We'll talk about that another time, and becomes water. Why is this important? The whole purpose is this is what keeps the electron flow going and keeps this whole process moving. Electrons start at fuel, get transferred over to a carrier, NADH, which transmits them to the electron transport chain, which gives them to oxygen, which becomes water. Electrons start at the fuel, namely glucose or another thing. They're handed over to NAD+, becoming NADH. NADH hands those electrons to the electron transport chain, which then hands them to oxygen, which becomes water. If you don't have oxygen, the whole process stops cold. Everything happening in the mitochondrion stops completely. And that is literally the reason you need oxygen in your lives. Without the oxygen, none of this process works. And you very soon afterwards will die from lack of ATP in certain tissues of your body. If your heart's not getting enough oxygen to keep this process going, it won't be able to pump anymore. If your brain's not getting enough oxygen, it won't be able to do this process anymore. And that is lethal. That is absolutely lethal. Okay, that's it for today. That is a whole lot of stuff in one video. Feel free to rewind and watch any more of this as you need to. I'm sorry for jamming so much into one video. I'll try to cover more important aspects of it next time. Thanks, everybody.